Hi everyone, thank you for being here. Thank you to the organizers for giving me also the opportunity to speak because I am really excited about sharing this project with you guys. I have to start also by saying that the format of the slides got a bit lost on the way, so I hope you can read everything properly. If not, we can discuss afterwards. Anyway, I want to talk about this intestine-on-chip approach to study fungal infection. And I know we just heard about Candida albicans, but I'm actually a microbiologist, so I still want to talk about it a little bit more. Candida albicans is a fungus, and you may know it because it causes candidiasis in women, but the main reservoir of Candida is actually not the vagina, but is the gut. Almost all of us in this room have Candida albicans in our gut, and we are not sick. It lives as a commensal, but we can get sick under some predisposing conditions, and these conditions have to do with already being weak. For example, being taking immunosuppressants, long-term antibiotic therapy, aggressive surgery, and in those cases, Candida albicans becomes a pathogen, and as you can see here, it will, is this working? Oh yes. It will develop these filaments, they are called hyphae, and these filaments are critical because Candida albicans will use them to invade through the tissue, it will cross the epithelial barrier, and it will translocate to the bloodstream, and there is when it can give the systemic disease, and in this systemic disease, many other organs become affected, and it can be lethal to many people. So it's a lot of us in the Candida field who are interested in this invasive candidiasis process specifically to be able to prevent it or to cure it. And we as a community, we have used mainly two models to study this. In vitro models are the most advanced ones, are based on transwells, and we have CACO2 cells in sometimes together with other goblet. And they have been really useful to dissect molecular mechanisms because they allow to zoom in a lot. And for example, many of the function of the genes of Candida have been discovered in these models. And they are also used to do drug discovery because they are high throughput. But for this specific case, we have the problem that they do not recapitulate the epithelial barrier, so we cannot really study how Candida albicans is going to cross it. So for more relevance in our field, we tend to use mice, which are great to study the systemic disease. And also commensalism, because these animals have features, like humans, that allow to keep Candida albicans as a commensal. They have the microbiota, they have the immune system, they have a tight epithelial barrier, but they have the main problem that Candida albicans is not a natural colonizer. So we have Candida albicans since birth, and this is not the case of mice. We have to force this colonization, which already comes with altering the system. And then, of course, we have the practical limitations, especially when it comes to dissecting the molecular mechanisms. This is why in our lab, in the Department of Microbial Pathogenicity Mechanisms, we decided to go for the intestine-on-chip to study the basic science on this. And I will not go into the infection model because Professor Mosik already gave a great overview. We are using also the biochips from Dynamic 42, it's the same model, but I still want to explain a little bit how we are using it specifically for the infection. We have the different cell types, we differentiate in circular perfusion for 7 days, and then to infect, we will change to linear perfusion. What we do to infect is perfuse Candida albicans in yeast form, the form that is commensal, and it will attach, and then we will constantly perfuse with new medium. What happens is that Candida albicans attach to the tissue and it forms this hyphae, and then these microcolonies. You have seen pictures before. What we do with this to analyze the host fungus interactions many things. We can, of course, take samples for a microscopy. In our case, we do this after fixing. We can also do, of course, molecular readouts, like the barrier permeability with fitsidextran. We collect the supernatants to assess LDH, the cytokines, and for us, it's also very relevant to plate those supernatants. We can see the amount of fungus that is not invading, just floating in the lumen, or most importantly, we can see the amount of fungus that is in the vascular channel, which means it would already be in the blood, it would already be on the way to systemic candidiasis. Finally, we can also collect the cell lysate and see how much of this fungus is invading or not. And this is a picture of how it looks like. You can see here the villi and the crypts of the host, the actin is polarized, and in yellow you have Candida albicans and the microcolonies I was talking about. Here there is a closer look, you see all the hyphae and how it's interacting with the host. And we had not actually been able to observe this in the other models we use, so we were interested in characterizing this. We did a time course. Here you can get an idea of how they grow. See, this is the yeast forming hyphae. At 8 hours, it starts branching, and at 12, we have the microcolony I was talking about. 16 hours post-infection, Candida is already growing a lot. And this is why we chose 12 hours post-infection for all our microscopy images. So all the pictures you're going to see from now on are at 12 hours post-infection. And well, besides looking at Candida, we are also interested in looking at the host to see how they are interacting. It's a great part of the infection, of course. So we were looking at tight junctions, and I hope you can see it, but we could see how Candida is disrupting the tight junctions. Here is the cadherin, where it's actually in contact, which makes sense because Candida albicans in the hyphae is expressing proteins that interact with the cadherin. We are also interested in looking at cell death. Unfortunately, we have been struggling to get this readout in a consistent manner, but at 12 hours we saw there is not much cell death, and the only dead cells that we see are here associated to the microcolony, which makes sense. For example, Candida is secreting, since very early on, toxins that will affect the cells. With this, yes, it is very nice to look at the pictures. They look pretty, but we thought that we wanted to actually get quantitative data from them. So this is why we established a collaboration with the Applied Systems Biology group at Leibniz HKI, where I work. And we together collaborated to develop an image-based analysis pipeline in which we can input these pictures and obtain lots of different data. So I'm going to explain you a bit how it goes. You have already heard a little bit about it, but it all starts with the original signal. We don't need much. We just need Candida albicans and the nuclei. And this is big pictures. They are tiles that comprise several or big parts of the tissue, and they are also in 3D, so they are chest tags. And with this, we can detect and reconstruct the microcolony, but not in 2D, but in 3D. This is trickier than it looks. Once we have the isolated microcolonies from each other, they can calculate all parameters about the morphology of the microcolony. It's something you want to know, they can calculate. And regarding the host, what they do is not only to calculate the tissue thickness, but they create a distance map. This means that for each point of the tissue, they can calculate the distance to the membrane. And this gives basically an idea of the topography of the tissue. If we overlap these two, we can get an idea of the host fungus colocalization and interactions. And all of this tells us how are the host and fungi looking like, but also how is Candida invading or where is it localizing. And for us, it's very important to know how changes in the environment are altering host and fungi. And we just saw this in Professor Mosik's talk, how Candida was looking different in presence of butyrate, for example. So I want to show something we have seen only with this analysis that it was actually surprising for us, and it has to do with this fungal invasion I was talking about. We, as you can see from the picture I drew before knowing this, we thought that the majority of the Candida would be actually invading the cells, but to our surprise, we found that only 20% of Candida was found under the cells, and 80% of Candida, here in blue, was found on the cells. This was surprising because Candida is a pathogen, so we would think that it's actually invading more, but it makes sense because it's easier to grow, just grow, than to invade the epithelial cells. Something that was also surprising was the localization in the tissue. We wanted to know that if Candida would have any preferred niches, and we would expect it would. So we separated or classified the tissue into high-density areas in dark, which would be villi-like structures in which there is a lot of distance between the tip of the villi and the membrane, and here low-density areas where it would be thin. And as you can see in this graph, there is a lot of variability that is probably depending on the donor. But also, we don't seem to see a preferred niche by Candida albicans, at least in our setup, so we think the places where the microcolonies are forming are probably depending more on where Candida gets to attach when we are inoculating it. And now, to continue, I want to also show you a project in which we are applying all these tools. We have done this together with Dynamic 42, and especially with Tim Cadden over there, who has done for this as much as I have. So what we are trying to do is to model the treatment of invasive candidiasis. So if a patient was in the hospital, in the ICU, and they were getting sick, they would be given an antifungal, they would probably be given caspofungin. This is administered intravenously, and we wanted to model how this course of treatment would be. So what we do is to infect our chip with either Candida albicans wild type or a clinical isolate that is resistant to echinocandins, which is the first-line antifungal used. What we do afterwards is to perfuse caspofungin in the vascular side, as it would be in the hospital, and we do this at concentrations that we have found in the literature, so what you would expect to see on a patient. After that, we can do microscopy, as I said, or we can also do molecular readouts for the host and the fungus. So what have we found? This is only some little snippets. When we perfuse caspofungin on the vascular side, we saw that the intestinal fungal burden is reduced, the fungal invasion is also reduced, and the translocation, so Candida going to the vascular channel, is almost completely prevented. We found actually one colony or one yeast across all our replicates, so at 4 micrograms per ml, which is a concentration you would expect to see on a patient, we would assume that this hypothetical chip patient would be getting cured. We also took pictures, and first of all, we were surprised that at 4 micrograms per ml of caspofungin, we could see no Candida at all, which was a bit surprising considering we had been retrieving fungal burden. And what we realized is actually that Candida was able to grow on the plastic of the chips, not on the cells, but on the plastic on places where the caspofungin could not reach. So we had to take this into account, but 4 micrograms per ml is actually a lethal concentration. At sublethal concentrations, as you see here, the colonies were looking a bit weird. So these are the pictures that went into the image-based analysis. And we found that at very low concentration of caspo, this volume of the microcolony was somehow increased, which was at the beginning surprising, but then we realized it also consistent with this called paradoxical growth that has been seen for other fungal species. And then we saw that at other concentrations, a bit higher, the colonies were less deep, so they would be less deep in set, and they were also more compact. This means that even if they are not dying, they are growing differently, which may alter how they interact with the host. We then repeated these experiments for the clinical isolate, and you can compare with the wild type in these blue lines. We saw that the fungal burden was only a bit reduced, even at higher concentrations, so we had to go up to 6 micrograms per ml. The fungal invasion was unchanged, and the dissemination, the translocation, was reduced, which makes sense, because at the end of the day, the caspofungin is in the vascular side, but it was still not completely prevented, and we found, every time we found colonies in the blood, which means that this patient would be still getting sick, and then we would have to actually rethink the treatment, think about other options or other concentrations. We also took pictures, and unfortunately we had not been able to analyze them yet because we did not have time before the conference, but I am actually really excited about doing it because we may be able to see something that we cannot see with our eyes. This is all snippets of a project we are actually trying to finish. We are trying to wrap up, and maybe you can read about it soon, hopefully, so stay tuned. And with this, I'm almost at the end, and my take-home message is that we have been able to study Candida albicans infection in ways we had not been able before. So, of course, we have the intestine-on-chip, and I don't need to tell you all the advantages, but for us it has been crucial to have an epithelial barrier with an immune system that can recapitulate more the host-fungus interaction in the body. And we have seen how Candida albicans is actually forming microcolonies and invading with them, disrupting the epithelial junctions and inducing cell death around them. Not only we can reproduce this invasive candidiasis, but we can reproduce how clinical antifungals are working and how some clinical isolates are resistant to these antifungals that we are using every day in the hospital. And in addition, we have the image-based analysis to ask and answer new questions and get additional insights. This has allowed us to, for example, see that in the chip, the lethal concentration that we see is way higher than the minimal inhibitory concentration that you would find in textbooks. This we are talking about, we saw 4 micrograms per ml, and 0.1 is what you would find in the manuals. At the same time, it's also much more similar to what we are seeing in hospitals, which means we are reproducing better than other in vitro models that we are using. Also, we saw that sublethal concentrations of the caspofungin are altering the microcolony morphology, which is associated with less pathogenicity, something that we should also take into account when designing treatments. And with this, I am truly at the end. I want to stress that this has been a really big collaborative effort with many people that know a lot. I want to especially thank my group of microbial pathogenicity mechanisms, my supervisors Bernard and Mark, the group of applied systems biology with Paras, Tubian, and Tilo, who have done most of the modeling, Dynamic 42 and especially team, and the Inspire group and Professor Mosik for the expertise in chips. Thank you so much all for listening, and I'm happy to take questions.
One very naive question about the cell death point that you have made. What is the mode of cell death when
So the Candida albicans is actually releasing a toxin that is a pore-forming toxin that goes inside the cells, and then the cells die because of the action of this toxin.
Okay, so that's why maybe you're using PI or something, but it's not real.
The thing is, we want to do cell death when the cells are also fixed, which means that they are also dead, which I know it sounds kind of complicated, but because we really want to stain Candida albicans, and if possible different proteins of Candida albicans, we really need to have the cells fixed, which is not very compatible with the current cell death stainings that are normally available.
Hi, thank you very much for your talk. I was wondering, so in your system, you say that the drug is introduced through the vasculature system, but since it's a gut infection, when in the hospitals, when they introduce the drug, is it through also the blood, or can they just not give a pill that the people get? Can it not be oral ingestion of a drug? So can't you just put the drug directly in the cell culture medium?
I don't know. So I don't know if they can, but as far as I know, they don't.
Okay.
So when you look at the caspofungin, it's mostly intravenously, and many of these patients are already in the ICU, very sick, and they would not even take a pill. So I guess this is the main reason.
Okay.
I just had a quick question, which I'm almost blanking on. If anyone else has a question, I can try to remember.
Hopefully I'm not overdoing it with questions, but compliments, very nice. The image analysis, of course, is a particular nice feature, but I was wondering, are you not also generating any wet lab readouts like biophysical measurements, biochemical measurements, and I think Alex alluded to some gene expression analysis. I think that would be cool and very valuable additional information apart from these pictures and the analysis of them.
Yes, so thank you for the question. This is a good point, and actually we started doing that even before the image analysis. We take supernatants to do the cytokines. I also did not have time to put them yet, but the cytokine profiles look very nice. We can see the inflammation and how the treatment is reducing it. We have also seen we can also see the LDH damage to the host. We could measure many things that I have not had time to show, unfortunately, but they are definitely there. Yeah. And gene expression we could do if there was something that we wanted to see specifically, which it may be in the next experiment.
Then I have a specific question to that point. Okay. Now, both here the host and the pathogens are eukaryotes. So how will you be able to distinguish the changes in the host cells from the changes of the fungal cells in the same co-culture?
So I understand the question, but I don't understand what you mean specifically with changes.
No, well, look, when a normal gut probably does have certain genes upregulated, others downregulated. By the moment there comes an infection, you can see these changes if you do RNA analysis and things like that. The same happens, you may assume, for the Candida. It changes from being commensal, then becoming hyphenated, then doing microcolonies. So actually you would like to see the changes in both systems, but I'm not a microbiologist, so I wonder how do you dissect the two that you can say that RNA change I'm observing is a host cell change or it's a pathogen cell?
Now I understand. I have not done this personally, but in our lab we do a lot of these dual species sequencing, and basically there is protocols that allow you to separate when isolating the RNA, for example, they allow you to separate the fungal cells from the epithelial cells because they have different properties. So this would not be a problem at all.
Yeah, I can ask the last question. So to your anomaly that you saw at 0.25, do you think that that could be linked to somewhat of a resistance already developing because these are clinical isolates? Do you think that you can somewhat dissect it?
This is a good question. I don't think so, based on that other fungal pathogens at lower concentrations of caspofungin, they also show these kind of weird bigger colonies. But as far as I know, this is not really a resistance mechanism, and we would, of course, have to look into it because it would be dangerous if these colonies were indeed more dangerous for the person, because when a person is taking antifungal, they are always going to have lower concentrations when it's being metabolized. But as far as I know, it has not been described that they are more dangerous. And we definitely want to look into it, but we will see. Thank you.
Thank you so much.